Good day everyone. So before we start, let me introduce myself. My name is Christine May Asuico. You can call me Ma'am Tin. Kaya wala lang guys, ganahan lang ko casual. Anyway, I will be your instructor for biopharmaceutics and pharmacokinetics na subject nyo this summer semester. And, ay my gosh, na problema na ko unsaon pagsakto sa atong mga topics, no? Pero ayun, let's try to cover all relevant topics under biopharmaceutics and pharmacokinetics. So, now let's start with our unit 1, which is the introduction to biopharmaceutics and pharmacokinetics. And as mentioned sa orientation, guys, I will require you to make notes regarding our discussion, and dili magmatter if typewritten or handwritten inyong notes, basta pagpasa mo og notes sa ako. And for this topic, in PDF form ang inyong ipasa nga notes. So, send lang nako ang drive link or ang folder kung aha mo mag-submit sa inyong notes. And this will serve as your attendance and assignment for this topic. So, let's start. So this will be our discussion outline. First is the definition of terms, particularly terms relevant to biopharmaceutics and pharmacokinetics. Then we will also discuss the Ladmer system, pero pahapyaw lang guys kay isa-isahon man nato na sila sa later topics nato. And lastly is the routes of administration, and pahapyaw lang po din guys kay I believe humana naman mo dinhi na topic, 'di ba, sa inyong DDS, pero ayun, i-review na lang nato. So let's start with our definition of terms. So our first term is the drug product performance, and before we dwell on that, i-review sa nato guys kung unsa atong drug or drug substance and drug product. So when we say drugs guys, these are substances intended for use in the diagnosis, cure, mitigation, treatment, or prevention of disease. So, kana nga definition na balik-balik na lang. So, I know, kabalo na mo ana. Pero, ayun, basically, when we say drug substance, we are referring na to our API or active pharmaceutical ingredient. Meanwhile, when we say drug product, this is different from our drug substance kasi itong drug product consists na ito of our API and excipients. So, itong drug product, ito na yung dosage form natin, such as tablet, capsule, unsa pa man, syrups, patches, etc. So, drug product performance refers to the release of the drug substance from the drug product, either for local drug action or for drug absorption into the plasma for systemic therapeutic activity. I think naka-encounter naman mo sa local drug action and systemic drug action na terms guys, no? Pero sige, review na lang. When we say local drug action, this is a drug action on the site of administration. So, for example, mag-apply ta og analgesic into topical na form. Then, sa skin man na siya ibutang. So, ang effect niya sa skin lang pod, and ana, basta local drug action. Meanwhile, when we say systemic drug action naman, the effect is in the entire body na, or to the entire body na siya, or on the site other than the site of administration. So, for example, naamoy, unsa man, antidepressant na tablet. Then, diba, gina-take na siya orally. So, murag ang iyahang general site of administration kay atong GI tract. Apil naman dara ang atong mouth, diba? Then, ang effect ana guys, dili man sa GI tract sa atong antidepressant tablet, 'di ba sa CNS man na siya mag-take effect. So ayun, kanang antidepressant tablet nato mag-undergo pa siya various processes para makaadto siya sa CNS. So that is an example of our systemic action or drug action. Now, For this concept, guys, what you have to remember is that drug product performance is related to our liberation na process. Di ba guys, if mag-inom tag tambal, usually dili man jud dayon siya mag-take effect. For example, paracetamol, if mag-inom ba di ay mo, mawala ba di ay dayon ng inyong kalentura, di ba dili man. That is because our drug, once it enters our body or once administered, mag-undergo pa siya o various processes para mag-take effect. And mauna na siya atong ADME nga i-discuss na to thoroughly later on sa atong subject. Now, para mag-proceed sa ADME ang atong drug, kailangan i-release siya from its dosage form. So, it is important to ensure na ma-meet nato ang expected nato nga drug product performance so that our ADME process will be initiated ultimately towards our desired therapeutic effect. So, inana siya sa atong drug product performance. Then, our next term is biopharmaceutics, and this is the study of the relationship between the physiological aspects of the patient and the physicochemical properties of the drug product. Then, another definition of our biopharmaceutics from our reference book is that it examines the interrelationship of the physical and chemical properties of the drug, the dosage form, and route of administration, and the rate and extent of drug absorption. So, in summary, biopharmaceutics studies the relationship between the physicochemical properties of the drug's biological fate in the body after its administration, or basically our ADME, and the resulting pharmacological action of the drug. Then, our next term is pharmacokinetics, and this is a branch of pharmacology that mathematically describes how the body affects the drug after administration to the processes of absorption, distribution, metabolism, and excretion. So, in simple explanation, I think na-discuss na ni sa inyo, especially sa pharmacology. But when we say pharmacokinetics, we are studying the effect of the body to the drug, or how the body affects our drug. So, inana lang ang basic na information about pharmacokinetics. But, in describing such, dili jud siya more on words. Basta pharmacokinetics, more on mathematical na expression, mathematical expression gina-describe kung unsa ang effect sa body sa atong drug. And in pharmacokinetics, more on rates atong ma-encounter dire. So, rates sa absorption, distribution, metabolism, and excretion, mga inana atong ma-discuss sa pharmacokinetics. And pwede po ta magamit og mga graphs, formulas here, and depends na lang sa parameter na atong i-discuss. So, we have different parameters to describe our ADME, guys, and we will discuss those in our later topics. Then, let's take a look at this figure, guys, no? So, in this figure, makita nato kung aha si drug product, and aha si pharmacologic effect. Pero, kani siya nga figure, guys, actually, this is a overview of our ADME na process. And let's take a look kung aha present ang atong drug product and our pharmacologic effect. So, let's start here kanang naay nakalingin dira nga red na box. So, that point is our administration or intake of drug na point. So, in this point, the drug in its dosage form is taken by the patient by an oral, intravenous, subcutaneous, transdermal, etc. route of administration. So, dinhian na to nga point makita si drug product. Again, dinhian na to nga point makita si drug product. Then, after that, our drug will be released from our drug product. And ang process na kay ginatawag nga liberation. And in liberation, ang final jud na product na to is our dissolved drug. So, dapat katong drug nga na-release from drug product, ma-dissolve siya sa atong body fluids. So, again, in liberation, there is a dissolution of our drug after the release. So, mauna siya ang atong liberation, guys, ha. And then, ni ang point na to, makita atong drug or atong drug na mismo. Then, after that is our absorption. And in absorption, yung drug natin, magpunta na siya sa ating systemic circulation or sa our blood. So, basta absorption, guys, remember lang na si drug, naa na siya or moadto siya sa atong systemic circulation. Then, once in systemic circulation, ma-distribute na ang atuang drug. Then, specifically, some fraction of the drug is absorbed from the site of administration into either the surrounding tissues for local action or into the body. Then, if into the body, guys, that is our systemic na effect, no? Pero, main idea lang ta, basta distribution, atong drug mo adto from systemic circulation, mo adto na siya sa atong mga tissues. So, depends na lang if tissues for local action or tissues nga na ay mga receptors, then, ano siya, kanang systemic na action. Then, once ma-reach na ni drug ang iyahang target na site, mag-elicit na siya og pharmacologic or clinical effect. Then, in this point, guys, ayun na pharmacologic effect. Or at this point, ma-reach or mag-take effect lang atong drug, basta ma-reach na to ang atong minimum effective concentration sa atoang site of action. Again, guys, pharmacologic or clinical effect will only happen if ma-reach ang atong minimum effective concentration sa site of action or ma-exceed ang atong MEC. So, then mahitabo ato ang pharmacologic effect. Then, after that, ang mahitabo sa atong drug from the tissue, mubalik siya sa systemic circulation para ma-eliminate or para ma-metabolize and ma-eliminate. So, ana ang murag overview sa atuang ADME. Sige. Our next term is drug concentration and drug response. And in this concepts, it tells us that the dose and frequency that a patient takes a drug is dictated by the manufacturer's recommended dosage regimen or the regimen prescribed by the physician. So, basically, this tells us na the amount of drug and unsa man and how frequent siya i-take by the patient is presented by the manufacturer based on the studies na gikonduct nila, and these studies are very focused on biopharmaceutics and pharmacokinetic actually. Then mauna na siya ang makita ninyo sa drug label na recommended dosage depending kung ano yung population natin. Then dili pud na siya ang atong final say, kay ang final say man jud kay ang physician nato kung unsa ang i-prescribe sa atong physician na dose and frequency sa drug, mao man jud na ang i-follow. However, due to individual differences in the patient's genetic makeup or pharmacokinetics, the recommended dosage regimen drug may not provide the desired therapeutic outcome. Guys, take note na yung mga data na makukuha natin from biopharmaceutics and pharmacokinetics na studies, these are data from the average of our population. So, average na siya. 'Di ba? What works for one person might not work for another. So, although okay ni siya kunwari nga dose and frequency ani nga population in general, naa jud tay mga exemption for that, and that is attributed to the genetic makeup of our patient and their different pharmacokinetic na profile. So for example, si patient A, if hatagan siya 500 milligram nga paracetamol, 80% ana nga paracetamol maabsorb dayon sa iyaha. Mao daw na siya ang kunwari, mao na siya ang normal na absorption percentage for our paracetamol na 500 milligrams. However, in another person, kay patient B, kunwari, if mainom siya 500 milligram nga paracetamol, ang maabsorb lang sa iyaha or ang maabsorb lang niya kay 50%. So, that is an example of difference in the genetic makeup or pharmacokinetics of our patient in terms of drug. So, in ana siya. Then, our next term is plasma concentration. And our plasma concentration, in simple terms, it is the amount of a drug present in the plasma at a given time. And kaning plasma concentration nato, guys, important nga i-measure ni siya kay it assess drug absorption. Then, particularly, it tells us how much of the drug reaches the bloodstream. Then, another is it also tells us, or it is important to monitor therapeutic levels of our drug. Then, another is it is important for dosing. Then, itong plasma concentration, this is very important to study our pharmacokinetics. And another is it is studied to detect drug interactions or issues. So, yun yung use ng plasma concentration natin. Then, another is it is used to measure, or it is measured to confirm whether the drug dose was subtherapeutic due to the patient's individual pharmacokinetic profile or was not responsive to drug therapy due to genetic difference in receptor response. Pero, if ako ang i-ask dire, guys, ang importance sa plasma concentration kay naatay range for that from subtherapeutic to toxic. Then, ang desired na to nga point, dara ang range kay ang therapeutic. So, ganahan ta if ang drug concentration na to kay naa sa therapeutic level, kay dara man ma-elicit ang atong pharmacologic effect. Then, dili ta ganahan nga subtherapeutic, kay meaning, dili effective ang atong drug. Then, dili po ta ganahan nga toxic ang level sa atong drug concentration, kay harmful na siya sa ato ang patient. So, ana lang sa plasma concentration. Then, our next term is pharmacodynamics, and dili ra kayo ta mag-focus diri, guys, ha. But, this is the relationship between the drug concentration at the site of action and pharmacologic response, including biochemical and physiologic effects that influence the interaction of drug with the receptor. So, our simple explanation here, basta pharmacodynamics, it tells us the effect of drug to the body. So, mauna ni siya tong pharmacology, no? So, din niya sa pharmacodynamics, madiscuss pod ang mechanism of action of our drug. So, basta, again, pharmacodynamics, it tells us, or it is the study of how the drug affects our body, and nag-focus siya sa atong mechanism of action. Then, our next term is exposure and response. When we say exposure, this refers to the dose and various measures of acute or integrated drug concentration in plasma and other biological fluid. But basically guys, basta drug exposure, this is measured by the plasma concentration. Then, another term is the drug response, which refers to a direct measure of the pharmacologic effect of the drug. So, kung unsa man ang effect sa atong drug, mauna na siya atong drug response. Sige, another term is our toxicology, which has different branches. So, first is our toxicokinetics. This is the application of pharmacokinetic principles to the design, conduct, and interpretation of drug safety evaluation studies and in validating those related exposure in animals. Pero, in simple terms, toxicokinetics is pharmacokinetics in the toxicology nga branch, and ana lang. Then, another term is our clinical toxicology, which is the study of adverse effects of drugs and toxic substances in the body. So, dinhiya guys, kaning clinical toxicology, mura siya pharmacology, pero focusing on the toxic effects of our drug. So, it discusses how drug affects our body in a toxic level. So, unsa ba ang mga toxic effects sa atong drugs? So, that's it for our toxicology. Then, naamoy, or sa mga second years, pag third year ninyo, maka-encounter mo aning toxicology, naamoy subject for this. So, stay tuned. Then, naapod, siguro ko, I believe, nanay mga naho mag-toxicology din. So, in ato siya. Now, let's discuss the measurement of drug concentration. Starting with the biological samples, kung asa ta pwede mag-measure of drug concentrations. So, we have our milk, saliva, plasma, and urine. Pero take note lang of the most important guys, that is our plasma. So, mao na siya ang gold standard na to when it comes to sample or biologic sample for drug concentration measurement. Then, ang mga methods din we have sensitive, accurate, for a while guys, okay. We have sensitive, accurate, and precise analytical methods, and chromatographic and mass spectrophotometric methods. Then kana siya, di na nato na i-elaborate ha, kay naa naman na siya sa QC. So, kana, main idea din the biologic samples na ginagamit nato to measure drug concentrations are milk, saliva, plasma, and urine. Then, ang pinaka ginagamit siya is our plasma. Now, in getting our biologic samples, we have invasive and non-invasive methods. For invasive, examples nato din kay sampling blood, or pagkuha o spinal fluid, synovial fluid, tissue biopsy, or any biologic material that requires parenteral or surgical intervention. Basta isipon lang ninyo guys, basta invasive kay gina-open nato ang body or ang material nga ginagamit nato para magkuwag sample kay naga-enter sa body. E nana, basta invasive. Meanwhile, our non-invasive methods include sampling of urine, saliva, feces, expired air, or any biologic material that can be obtained without parenteral or surgical intervention. Then now, let's discuss the drug concentrations in blood. Pero, ang i-focus na nato diri guys, kay ang reason kung anong ang blood component na ideal jud for drug plasma concentration na pag-measure kay si plasma. Anyway, let's start with our whole blood. So, when we say whole blood, this contains cellular elements, and this is obtained by venous puncture and contains an anticoagulant such as heparin or EDTA. Then, our serum naman, Para makuha na siya, the whole blood is allowed to clot and the serum is collected from the supernatant rather after centrifugation. Then our plasma naman, this is obtained from supernatant of centrifuge whole blood to which an anticoagulant is present. Now, dinhi sa tulo guys, ang ginagamit jud to measure plasma or drug concentration rather is our plasma. That is because it contains proteins. And kanang proteins guys, nagabind din ha ang atuang drug. That is why ideal nga si plasma ang gamiton kay naatay ma-measure nga drug nga kato nakabind sa atuang protein. Sa serum manggod, wala nay protein dara guys. Appeal to siya sa nag-coagulate or nag-clot most likely. So sa serum walay protein. So wala pud moy ma-detect nga drug concentration din ha. Then if sa whole blood naman naay protein pero daghan pud siya other elements, so lisod i-detect ang atong drug dara. So that is why plasma ang pinaka-ideal nga sample for drug concentration measurement. Then our next concept is the plasma concentration time curve. And our plasma concentration time curve is a graph that shows how the concentration of a drug in the plasma changes over time after administration. And if mapansin ninyo din na sa graph, ang naa sa x-axis kay ang time, then ang sa y-axis naman, that is our plasma or drug concentration. Then our plasma concentration time curve has different components and kana mga naka-label dira. Then ano na to na siya i-define, pero dili na to siya i-discuss thoroughly ha, kay I believe humanan naman na siya sa FEFARM, pero kana i-review na lang nato. So our first term is the MEC or minimum effective concentration, and this reflects the minimum concentration of drug needed at the receptors to produce the desired pharmacologic effect. And diba na-mention ko ni, dapat ma-reach na to ang MEC para na ay pharmacologic effect, ma-reach siya or ma-exceed dara sa MEC ang concentration sa drug present sa atong site of action para na ay pharmacologic effect. Then, our next term is our MTC or minimum toxic concentration, and it represents the drug concentration needed to just barely produce a toxic effect. Then kanang MTC guys, most likely na siya or mas taas na siya kaysa sa MEC nato. Then our next term is our onset time, which is the time required for the drug to reach the MEC. Then next is our intensity, which is proportional to the number of drug receptors occupied, and it is reflected in the observation that higher plasma drug concentrations produce a greater pharmacologic response up to a maximum. Then next is the duration of action, which is the difference between the onset time and the time of drug to decline back to the MEC. Next is our therapeutic window, which is the concentrations between the MEC and the MTC. The next is our therapeutic index, which is the ratio between the toxic and therapeutic dose. So, basta therapeutic index, guys, murag ang general formula niya is TD50 over ED50. Then, ang gusto na ko i-emphasize diri guys, kay if taas si therapeutic index, meaning safe si drug i-take. Kay dako ang difference between sa toxic dose and effective dose. So, if kunwari, inyong effective dose kay 50 milligrams and anak, then Ang toxic dose na to kay 100 milligrams. Diba layo ang agwat nila? So, if masobrahan nimo gamay ang effective dose, dili siya mag-matter, dili siya maging toxic sa patient. Meanwhile, if baba naman CTI or narrow siya, meaning less safe ang inyuhang drug. Kay gamay lang ang difference sa toxic and effective dose. So, if ma-exceed lang na gamay yung effective dose, maging toxic na siya sa ato ang patient. So, ana ang murag concept sa atong therapeutic index. And I hope na-discuss na ni siya sa inyong PCOL. Then, next is our peak plasma level, which is the average rate of drug absorption. The next is the time for peak plasma level, which tells us the dose, rate constant for absorption, and elimination constant of the drug. Then lastly is the area under the curve, which is the amount of drug absorbed systematically. Then kani siya guys, kaning area under the curve. Later on, we will discuss how to solve that one. So kana ha, take note of our mga parameters or parts of the plasma concentration time curve. Dapat kabalo mo ha kung asa siya. So, kunwari kani nga point, this is our minimum effective concentration. Then, kani siya, this is our onset of action. Then, kani, duration of action. Then, kani is our peak concentration. Then, this one is our minimum toxic concentration or MSC. Then, between that is our therapeutic range. So, ayun. Study this one, ha. Screenshot na. Okay. So, next, let's discuss the pharmacokinetic modeling. So, ano ba yung pharmacokinetic modeling or modeling? So, drugs undergo many complex processes in the body. They are absorbed, distributed, metabolized, and excreted, all potentially at the same time. And, lisod kaayo na siya i-monitor. So, to make sense of the overlapping events and predict how drugs behave over time, we use mathematical models. And these models help design drug regimens, predict drug concentration over time, estimate drug accumulation, correlate concentration with effects, and compare different drug formulations, especially in terms of bioavailability. Now, our pharmacokinetic model is a hypothesis expressed in mathematical terms that describes how variables like drug change over time, or drug concentration change over time. Then take note of our key definitions or term here. We have our independent variable, and in the case of pharmacokinetics, this is usually the time. Then we also have our dependent variable, and in the context of pharmacokinetics, this is usually the drug concentration. Then lastly is our parameter, which is a constant estimated from data. For example, elimination rate constant na to. Then, we have different types of pharmacokinetic models, guys. We have our empirical, physiological, and compartmental. And dili kita magdual din ha, guys, no? Pero kana mag-overview na lang ta. So, when we say empirical model, this is a mathematical model developed by fitting drug concentration data without directly reflecting real physiology or anatomy. Then, it focuses on describing the data, not necessarily explaining how the drug moves through the body. Then, next is our compartmental models, and in this model, ito yung pinaka-common na ginagamit, and in here, the body is represented as one or more compartments, not specific organs, but conceptual spaces where the drug distributes. Then, in here, naatay mga rate constants guys sa compartmental models. Then, we have two types of compartmental models. So, we have our one compartment and two compartment models. Then, in one compartment models, particularly basta IV, the drug distributes instantly throughout the body. Mauna siya ang assumption sa atong one compartment model na IV. Then, in terms of absorption, ang assumption nato sa one compartment model is that oral drugs first go through the gut or our GI. Then for two compartment model naman, ang gina-assume nato is that drug distributes between central, which is the blood, and peripheral, which is the tissue compartment. So in ana siya ang murag overview sa atong compartment models. So kana in ana lang to, same lang ang idea. Then kani siya words lang na siya. So basahon na lang nako no. In one compartment model, the drug distribution is considered instantaneous throughout the entire volume of distribution, and the body is considered as a single, kinetically homogeneous unit that has no barriers to the movement of a drug. Then, in two compartment model, central compartment, or there is a central compartment and peripheral compartment. Then again, our central compartment is the blood. Then sa peripheral compartment naman, that is our tissue. Then, drug distribute throughout the body at different rates. Then, particularly, first, Joyd, si drug daw, ma-distribute siya sa highly perfused na area. So, katong area nga daghang veins, daghang blood. Then, next is sa mga tissue na which has limited na perfusion. So, that's the overview for our compartment models. Then, for physiologic naman na model, these models are more complex and based on actual anatomy and physiology, and they consider organ sizes, blood flow, and tissue composition. And these are used to predict drug distribution and clearance in various organs and are particularly useful in drug development and interspecies scaling. Then, dinis sa physiologic model, no, daghan kaayog parameters nga gina-consider jud. So, for example, when talking about absorption, gina-consider si solubility, positive permeability, and active transport na present sa ato ang site. So, daghan kaayo siyag mga sub-parameters, no? So, basta, very complex si physiologic model. And if you want to learn more about our pharmacokinetic models, pwede ninyo siya basahon sa reading material nga isend ko. Then, naapod na siya sa book na to. Pero ayun, very tedious na niya siya. So, let's not dwell on that so much.